Hi, everyone. My name is Elijah Pantoja. I am the annual giving coordinator here at Pitzer. I've been at Pitzer since November 2019. I'm also a graduate of the class of 2018. So I'd like to start the session by introducing our speaker, Professor Patrick Ferree. While I didn't personally take any classes in Keck, I've only heard great things about you, Professor. So I'm excited for this discussion. But Professor Ferree is an associate professor of biology at the Keck Science Department. He has been with the college since 2010. He received his PhD from the University of California, Santa Cruz, and his postdoc at Cornell University. His recent courses include introductory biology and developmental biology. His research interests are chromosome structure and function, heterochromatin and satellite DNA, and mechanisms of microbial and genetic parasites. Thank you for joining this session titled Hijacking Reproduction, the Costly Antics of Selfish Genes, Chromosomes, and Other Components of the Cell. Please join me in welcoming Professor Patrick Ferree.
Thank you. Yeah, sorry, Elijah. We didn't cross paths when you were taking classes here. It's good to know that students are saying at least okay things about me. Thanks for attending the talk. And I'm going to go ahead and share my screen if that's all right. So Elijah read out my the first version of my talk. So that was Hijacking Reproduction. And I originally was really ambitious to think about talking about a lot of different things like selfish genes and chromosomes and other components of the cell. And I've underlined other components of the cell because this is a revised title. And what I really thought as I was planning this talk was you can spend a seminar or two on each of these groups of things. And so I'm really going to focus on bacterial symbionts, which is a lot of what my research is centered on. So we're gonna focus on that. And I'll start out with a slide that depicts this illustration. It depicts a scene, I think, in London back in the 1600s during the Bubonic Plague. And the plague is caused by a bacterium called Yersinia. And this is a micrograph, a fluorescence picture of Yersinia bacteria that have been cultured in the lab setting. But bubonic plague was terrible. Millions of people died. And apparently there were different waves of the plague. There was a big wave in Europe in the 1600s, and then there were other waves. So it was a terribly lethal disease caused by this bacterium or different strains of this bacterium. One of the main ways that the plague was carried, that was crossed between person to person, is by the flea, this little insect, a pesky insect. And so when I was reading up about this, it's really interesting because the bacterium Yersinia lives in the gut of the flea. And apparently the bacterium makes what's called a biofilm. It's a little matrix of proteins and carbohydrates. And that causes inside the flea's gut tube, it causes the flea to regurgitate. So what the flea does is it bites a host. It can bite rats, and it's carried by rats, one of the many different vectors of fleas, but it also bites humans. And so let's just imagine that it's carrying Yersinia in its gut. And so if it, in a rat infested area, if you get bitten by one of these fleas, the bacterium inside the gut of the flea actually makes the flea bite harder. And then when it bites, it regurgitates the blood meal of the previous whatever it's fed on. And that regurgitation, that is like a vomiting reflex, actually causes more bacteria to be spewed into the bite of the victim. So it's a self-selecting mechanism for the bacterium to sort of hijack this flea in order to transmit Yersinia, to transmit the bacterium more effectively. This is an example of a bacterial pathogen, a disease caused by a bacterium like Yersinia that's transmitted what we call horizontally. It's a horizontal mode of transfer. And what that means is that if you have an infected person in the middle here, This just symbolizes a population of people. And there's a transmission, a horizontal, across the different people from person to person, from the infected to uninfected. That's what's meant by horizontal. In this particular case with the plague, there could be other vectors, but there's a sidestep here, such as with a rodent, and I'm not showing a flea in this diagram, that would be pretty small, but the point, I mean, the transmission is across the population of people. A lot of other horizontally transmitted bacterial pathogens that cause really nasty diseases. Mycobacterium causes leprosy and tuberculosis, syphilis, anthrax, scarlet fever, diphtheria, typhoid. They're all caused by different bacterial pathogens. And in each of these cases, it's a horizontal transfer. There could be an agent that helps to transfer, but in most of these cases, these bacteria are horizontally transferred. And I put this slide in here because it gets across, other than, let's say, a rat that carries a flea and you get transmission by being bitten by the flea. There are a lot of other ways that these bacteria can be transferred horizontally from person to person, through saliva, through sneezing and coughing, something as simple as a handshake, tainted water. If you aren't careful, you can pick up some pretty bad bacteria from dirty water, and then contaminated food and uncooked food. Okay, so that's horizontal bacteria. And what I'm going to focus on from now on is a different class of bacteria. Some of these bacteria, all of the bacteria that I'm going to talk about, that is some of the bacteria in nature, are transferred vertically. So it's not across person to person in a population. It's really from parent to offspring. These are bacteria that are inherited from parents to offspring. And then when those offspring grow up, if they have kids, they transfer the bacteria to their kids. So vertically transmitted bacteria have been well studied in insects, especially in arthropods in general, but especially in the insects. And they're very, very common in the insects. And some names that I'm going to mention through the next couple of slides that fall into this class are Wolbachia, Cardinium, Arsenophonus, and Spiroplasma. And so these are some of the more well-studied bacteria that are vertically transmitted, and their hosts are insects. So all of these bacteria, these four that I just mentioned, and others, are not just parentally transferred vertically from parent to offspring, they come from mom, from the mother to offspring. It's a lot like mitochondria. Does anybody know what the mitochondrion is? Yeah, so it's a symbiont that used to be a bacterium. It's an organelle in all of our cells, and all eukaryotes have them. It's a characteristic of eukaryotes, and they produce cellular energy. If we don't have them, we couldn't exist. So they are, in effect, ancient bacteria that were engulfed by another cell, and that is the framework for the eukaryotic cell. So these bacteria are maternally transmitted exactly like the mitochondria. So they're transmitted through the eggs of the mother to her offspring. Okay, so why are these bacteria vertically transmitted? Why are they transmitted through the mother, the female lineage? So the way people study this is that you can take an insect. This is a lot of what we do. We work with different insects in my lab here in the Keck Science Center. But let's just say you're taking a female insect and you can dissect out her ovaries. So these pear-shaped organs here, you can take them out, and this is a real picture of ovaries, really nice big ovaries here. And all of these little units are individual mature eggs that are ready to lay. And so the female, if she's gravid, she would just push those eggs out if she finds a suitable place to do so. And if you look at the individual eggs as they're developing, now these are mature, but at the very tips here are immature eggs. And so if you look at those under the microscope and with special stains, here is a developing egg. There's a nucleus, and this green stuff are intracellular symbionts or bacteria. So a lot of these bacteria, you can't really think of them as just as free living bacteria because they live obligately inside the cells of these females' tissues. Okay, they have to live there. And let me just say, if you're going to be if you live in cells and you're going to be transmitted from one individual to another, the egg is the best place to do that. Because number one, the vast majority of the other cells in the body are never transmitted. They're part of your body, right? The reproductive cells are the key to getting to the next generation. And let me just say, eggs are much better transmitters of bacteria than sperm. Sperm are teeny tiny. They really don't have any space in them at all other than a little nucleus. So they're terrible transmitters of bacteria. So if you're a bacterium and you live inside cells and you want to be transmitted, you need to be in the eggs in order to make it to the next generation. So just to echo what I'm saying, what I've already said, these bacterial symbionts, and I call them symbionts because they're really confined to the cell and that's where they live. So they have a symbiosis with the organism that they live in. They do some really crazy things in order to be transmitted. And that's what I want to focus on from now for the rest of the talk. They manipulate the host reproduction. So if you think about horizontally transmitted bacteria, let's just say there's a bacterium and it infects the nasal cavities. One way that it could be transmitted is it makes a person sneeze, and then the particles of the sneeze actually come into contact with someone else, right? But if you're vertically transmitted, how would you help the host to transmit you? And it turns out that a lot of these bacteria do some really wacky things with the reproduction of their hosts. And I'll talk about that in a second, but in general, these manipulations that they do ultimately increase the number of females at the cost of males. So they are selecting for females, or they just selectively favor the females. So they want to give benefits to the females because the female lineage is how they survive. And if they can help the females, that means if the females can produce more babies, that means that they are going to do better in nature. Ultimately, why are they doing this? The females and not the males are the transmitting sex. Again, because the egg is the best place to be if you want to be transmitted to progeny. Okay, so I'm going to talk about 4 different ways that these bacteria, different types of symbiotic bacteria, manipulate reproduction to benefit themselves. Some of these are kind of wild. So the first is called feminization. And has anyone ever seen this little creature? A little picture of, I had a name on here, I guess it disappeared. Yeah, it's called, well, yeah, my kids call them pill bugs or roly-polies. They're all over the place here in Claremont. Yeah, so there's a bacteria called Wolbachia. I had mentioned the name before, and I'll say more about Wolbachia later. But Wolbachia infects these insects among many others. It's actually an arthropod. It's not a true insect. But what Wolbachia does in this species is this is a male, this little kind of copper colored one, and the smaller, that's a male, and this is a female. But they're really both genetic males. So if Wolbachia is present, it causes the transformation as this animal is growing up, as it's developing, it causes the transformation of this male into a functional female. And that's what I'm symbolizing here. So you have a genetic male, it's got the sex chromosomes that say you're going to be a male. And then there's a transformation, we think through hormones during development, to cause that genetic male to become a functional female. So it develops ovaries, it has the mating behaviors of a female, and basically that female is Wolbachia infected, and it then, because Wolbachia is transmitted through the egg cytoplasm, she mates with, let's say, an uninfected male, and all of her progeny will be Wolbachia infected. And then the males, at a certain period in development, they will be transformed into females. So this is called feminization. Another manipulation that these bacteria do is called parthenogenesis. A colloquial term for this is virgin birth. So this is essentially when a female can produce, in most cases, it's a female producing all female broods, all female progeny, without the need for male fertilization. So the males are not needed at all in the case of parthenogenesis in a lot of species. I just put a couple of insects here that we know has parthenogenesis. And again, it's because of this little bacteria called Wolbachia. Now, the strain that causes feminization that I just talked about in roly-polies is a different strain of Wolbachia than is causing parthenogenesis in this little wasp called Trichogramma and another wasp called Mucidiferax. Okay, there are other insects that have parthenogenesis too. The unsure thing is whether Wolbachia is causing parthenogenesis, or could be other symbionts that are also causing parthenogenesis. But essentially, if Wolbachia is in a female, there is a cellular process that causes her to produce all eggs that are going to eventually develop into females. So they never develop males. So it's different than feminization. Feminization, a female will produce early males and females, and then at some point in development, the males will transform into females. Here, it's just that the reproduction has been hijacked so that they produce all female broods. And again, they're all Wolbachia infected, and they won't need fertilization from males from that point on. As long as this bacterial strain is in them, they will not need males. And in fact, there are some species of Mucidiferax for which there are no known males in nature. And all of the females that we know of are infected with Wolbachia that cause parthenogenesis. So there are evolutionary implications here. The bacteria is really influencing the evolution, we think, of some of these species. The third manipulation that I'd like to talk about, and I'll spend a little more time on the last two. This third one is called male killing, and it's pretty insidious. So let's say here's a female and she's infected, and let me just say, it's not just Wolbachia, certain strains of Wolbachia that cause male killing. There are other ones called Arsenophonus and Spiroplasma that also can cause male killing. So I'm just going to put infected here. So imagine that you've got an infected female with any of these strains of bacteria, and there's a mating. And when this female lays eggs, all of her progeny are going to carry that bacterium, again, because the bacteria is transmitted to all progeny. Okay. During development, any individuals that are genetically males are actually going to die. So there are different ways that these bacteria sense maleness or femaleness sometime during development. And in any of these cases, there has to be a male-specific process, a developmental process that's targeted. There has to be some cellular difference that any of these bacteria senses in order to kill males, but it does nothing to the females. And it's very powerful. So I'll say a few things about some work that some of my research students have performed a couple of years ago, actually about 5 years ago now. So we had been studying male killing caused by this bacterium called Spiroplasma, and we use, as a model insect, we use Drosophila. So probably most of you are familiar with Drosophila. It's the common fruit fly, and it's used by lots and lots of geneticists. It has been used over the past 100 or so years, and people still use it a lot for genetic experiments. And so Spiroplasma causes male killing in the fruit fly, among other species, and so we can use this whole system, Spiroplasma and the fruit fly, as a nice way to sort of figure out what are the aspects of how male killing works. We really want to understand what is the mechanism. How is it that the bacterium targets males so that it kills them deader than a doornail while leaving the females alone to develop? And so basically what my students did was they just looked at embryos. So these are embryos, early stage, and you don't have to be an expert in order to see what I'm going to point out to you. First of all, let me just say that. So these are just early, like right after fertilization, maybe an hour after fertilization, these embryos. Here are females and here are males. And these little red spots, maybe it's a little hard to see, but we can use it. We have a marker. We call this chromosome painting, but we know these are male females, and we know these are male embryos and these are female embryos. So we know exactly what we're looking at. And we know that all of these are infected because we have a spiroplasma infected Drosophila line. And if you look early in embryogenesis, you can see that these embryos look pretty much the same. They look fine. They're developing normally. About middle stages of embryogenesis, they still look okay, but the males are starting to look a little bit weird. Now, if you squint your eyes, like if you're looking up at the sun, you can kind of see a little bit of disheveledness here. Not so much, but toward late embryogenesis, it's probably a little clearer to see that this embryo looks really bad. It just looks irregular. You don't need to know exactly why. You could just see, wow, that looks like a really misshapen embryo. The females look fine. They're nice and segmented here. Those are going to be the body segmentations of the adult when it reaches adulthood. The males, you can hardly make out the segmentation. And there's a lot of asynchrony in the cell size and shapes and all that. Spiroplasma makes the males look really bad in embryogenesis. So we wanted to know why, like what is going on? And ultimately, we wanted to know what is the target of spiroplasma caused male killing. And it really there has to be some target that is male specific. It has to be something that males have and females don't, or vice versa. And so in males, like most insects, same is true for humans, females have 2 X chromosomes and 2 chromosomes at every other position. So humans have 23 pairs of chromosomes, and females have 2 X chromosomes and 2 chromosomes of every other chromosome position. Males have 1 X chromosome and 2 of every other chromosome position. So really at the chromosome level, if you're talking about fruit flies or you're talking about humans, females have 2 X chromosomes and males have 1. I am not showing the Y chromosome here because there's hardly anything on the Y chromosome. I don't know if you know that or not, but essentially there's at least one gene that I know of on the Y chromosome that is needed to confer maleness in humans. In fruit flies, it doesn't confer maleness. And so there can be males that don't have Y chromosomes. And anyway, that's why I'm showing X chromosomes here. But there are thousands of essential genes on the X chromosome. And those genes are needed for both males and females. And there's a problem. In nature, there's a problem with a difference in sex chromosomes that has to be solved. So if you think about it, let's say there are 1,000 genes on the X chromosome, and there are 1,000 genes on this other chromosome, some other chromosome pair. So all of those genes on the X chromosome are in 2 doses because every gene that's on this chromosome are also on this one. Does that make sense? Okay, same is true for here. So if we were going to represent the gene dose, the amount of product made per gene from a gene on the X chromosome and a gene on some other chromosome, these arrows would be about the same height because there's 2 copies of this gene on the X and there's 2 copies of these other genes on this other chromosome or any other chromosome. So that represents the gene dosage. It's kind of a 1-to-1 ratio. But in the male sex, it's different because males have 1 X chromosome, they only have half the gene dose. They only have half the number of genes as what you would see in females. Now, a lot of genes are produced on the X and on other chromosomes need to work together in order to make things function properly in the cell. So there's a dosage problem here. If you have gene dosage that's half as much relative to another gene dose on another chromosome in males, but you really need this is a problem that has to be fixed. And so the way nature has solved this problem at least in Drosophila and fruit flies, is called dosage compensation. There's a compensation mechanism that upregulates the amount of gene dose, the expression of genes on the X chromosome, and it bumps it up to a level that you would see in females. So this is dosage compensation is a male-specific process. And the way it works, there's a lot in this slide. I don't get caught up in the details, but essentially in males, there is an early signal that causes a dosage compensation protein complex. It's mostly proteins that forms. It does not form in females. And when it forms in males, it's sticky. It localizes to, it sticks to the X chromosome and remodels the hereditary material in such a way that it upregulates those genes. This does not happen in females because it's not needed in females. And you can make it happen in females, and if you do, it kills the females. Okay, so that's the way nature has solved this problem of dosage compensation, that difference between males and females. Well, we suspected that spiroplasma might be targeting this process. I put this slide in here because this is a really pretty picture showing some chromosomes, that there are different ways that my students can take out certain tissues from fruit flies and squash them. You've probably seen karyotypes of human chromosomes before, maybe. This is a karyotype of a fruit fly. The chromosomes are a little bit hard to read here, but that is the single male chromosome, that is one X chromosome, and you can see that it has been painted with this dosage compensation machinery. It's pretty red signal here. Okay, so it just shows when the dosage compensation machinery is made in males, it gravitates to, it sticks to the X chromosome where it can make those changes in those genes to cause them to go up. Okay, so our question was, does spiroplasma tinker with, does it inhibit somehow this dosage compensation machinery in order to mess up male development? And so the upshot is yes. And this was done by students actually, so actually a bunch of students, Tricia Chung and Jennifer Martin and Nithin Kupanda and Becky Cheng. So about 4 students had hands in these experiments. But basically, these are cells. You're looking in a cross section of an embryo here, and all you need to know is these little red and green spots. Those are probes. Those are little molecular tools that we can show the dosage compensation machinery, especially this red, sticking to the X chromosome in these embryos. And this embryo on the left does not have spiroplasma. It is a plain old uninfected male. And this is what's supposed to happen. However, on the right side is a spiroplasma infected male, and there are hardly any red spots. And so what happens is spiroplasma is somehow causing the dosage compensation machinery to not stick to the X chromosome. And in fact, we know that those little spots, you might be able to see those little spots here, are not the X chromosome. There are other places, there are other chromosomes, and that is not what's supposed to happen. As a result, the genes on the X chromosome become misexpressed, and the male embryos develop improperly and they die. So really what the question is now is how is it the spiroplasma causes this effect? That's really what people want to know. And recently there's a group I think in Switzerland, a geneticist who has discovered a gene that they think actually is the toxin that causes this effect. So now the question is, well, how does that work? And so different groups are working with this gene and trying to figure out how it acts as a toxin. Okay, the fourth and last manipulation that I'll tell you about is a little bit more complicated. So if I say something and you're like, I don't really think I follow it, please stop me and let me know, and I'll try to rephrase. So this manipulation is primarily caused by Wolbachia. This is yet another type of strain of Wolbachia. And I didn't say this before, but now's a good time to say it. Wolbachia may be the most widespread bacterial symbiont in nature. So people have done studies that show that at least 70% of all insects, and believe me, there are lots of different insects in the world, that over 70% of insect species are infected with Wolbachia. So this manipulation that I'm showing you is actually really widespread. This is one of the most common things that it does to its insect host. And it's called cytoplasmic incompatibility. And essentially that's kind of a mouthful, but really it's a conditional male sterility. And so I'm going to spend a minute or so telling you how it works. So imagine a cross where you have a male and he is infected with Wolbachia. And he mates with a female that is not infected. So I said this is a conditional male sterility. So the Wolbachia is actually going to make this male sterile. And here's what happens. Here's the basis of the sterility. This is a picture right after fertilization, and you should have the two chromosome sets. The sperm goes into the egg, and it forms a nucleus, and it undergoes one cell division, and you should have two nice round nuclei there. Well, you have three, and that's not normal. And this thing in the middle that's pointed by this white arrow is actually the nucleus that came from the sperm, and it's damaged. It's been tainted somehow by the Wolbachia such that it can't participate in that division. It gets stuck there in the middle, and these nice little nuclei here, they're fine, and they came from the egg. So Wolbachia has poisoned the sperm's nuclear material, and it becomes ejected, it becomes eliminated, and it'll get lost. But there's a problem here, right? Most species need a contribution of chromosomes from the dad and from the mom. I mean, humans are the same way. And so by killing half of a genome, that causes the embryo to eventually die. It doesn't really develop that long. And so this is the basis of this conditional male sterility. It's conditional because there's another scenario. If you take that same male that has Wolbachia, and so this is the male whose hereditary material in the sperm would be poisoned. If he mates with a female that has Wolbachia, that same Wolbachia strain, that poisoning effect is erased. So there's like a toxin and antidote effect here. The Wolbachia in the male toxifies, it poisons the sperm's DNA. Well, it's more than DNA, it's the nuclear material, I'll say. But that same bacteria, if it's in the egg, rescues or it reverses that toxic effect. It's like an antidote, okay? And that embryo is just fine. So what does this mean, okay? So at the population level, this is a diagram. I didn't make this particular diagram, but I think it's kind of nice. So here's a cross, and this is just two plain old insects. There's a female and a male, and neither one of them has the Wolbachia in this case. They're just uninfected, plain old flies. They're going to have a lot of babies. Flies are prolific with making babies if there's fruit for them to eat and whatnot, okay? But now if you bring Wolbachia into the picture, if this uninfected female mates with a male that he's shaded red because we're symbolizing him as to carry Wolbachia, that conditional male sterility effect that I just showed you, where the sperm's the sperm's hereditary material is eliminated, it kills all the embryos. They're all completely dead. They never develop. So that's a very strong effect. But if the female is infected with that same Wolbachia strain, she can mate completely successfully with that same infected male, and all of her progeny live because the antidote effect has kicked in. And by the way, infected females can mate just fine with uninfected males because there's no toxic effect there. So if you just take a look here and see, Wolbachia infected females are very privileged. They can mate with any male in the population. But if you're not infected and you're a female, you can't mate with infected males. You can only mate with these. And what this does is this eventually ratchets up the amount of Wolbachia infected individuals in a population, because the advantage is all for the infected females. And the more infected females lay babies and they're all infected, you're going to have more and more Wolbachia, more and more Wolbachia infected males, more and more Wolbachia infected females, and these are going to eventually die out. And it's actually a very rapid effect. So this is a self-selecting mechanism that the Wolbachia is using to manipulate the reproduction of these insects in order to selectively benefit themselves, to increase their frequency in nature, and it works really well. Okay, so why does this matter? Well, there's a disease link here, and it's tied to humans. So there are several diseases that are caused by things like filarial nematodes. So here's a filarial nematode. I don't know exactly which species this is, but it's one of the many that are actually carried by mosquitoes. So this is an adult worm, but the nematode as a juvenile at one of the early larval stages actually is transmitted in blood and can be transmitted by mosquitoes. Okay, and there are a couple of diseases that are caused by the nematode. One is elephantiasis and another is river blindness. And I've actually seen cases of elephantiasis when I was visiting relatives down in the Dominican Republic a couple of years ago. So it's even in this part of the world. The nematodes, interestingly, the link here is that it's not just arthropods that carry bacteria, that carry Wolbachia. These nematodes actually are obligately infected with Wolbachia. Obligately is underlined because that means they all have Wolbachia, and the Wolbachia is needed as a true symbiont. It is needed for these worms to live and develop. So you can actually kill the filarial nematode by taking someone with elephantiasis and giving them certain types of antibiotics. So that kills the worms. And that's good, except for there's been a lot of, number one, there's been a lot of damage by that point. If you know you have elephantiasis, there's been a lot of damage to your lymphatic system. But it also, when you kill the Wolbachia, you kill the Wolbachia with the antibiotic that kills the worms. Apparently there's a really bad inflammation reaction that can exacerbate it, can worsen the effects of the disease, at least for a while. So it's really it's a really rough situation. Mosquitoes also carry viruses that cause pretty bad human diseases. I've shown a couple of different mosquito species here. Some of those are actually in the LA area, unfortunately. And they carry things like Zika, West Nile, and dengue. And so there's a lot of interest now in actually using Wolbachia as a way of controlling these mosquitoes that carry these really debilitating diseases. And a lot of these are neglected diseases. They're not some of the high profile diseases that there's a lot of NIH money being pumped into to solve or to fix. But it's becoming more and more common now that people are putting their attention into controlling mosquitoes. And Wolbachia has been on the forefront of some sort of natural way of combating mosquito populations. So in a lot of different countries, and I think I have a slide, the next slide I'll show a couple of places where projects like this are taking place, but a lot of governments in other countries are putting a lot of money into infecting certain mosquitoes with various strains of Wolbachia, like a strain of Wolbachia that actually causes that conditional male sterility. So you can use microinjection in order to infect these mosquitoes. You grow these mosquitoes up and you mass produce them. So there are insectaries that are producing large amounts of mosquitoes, and that can be released into the wild. And so there are a couple of objectives here for doing this. One is that it really has to do with the fact that Wolbachia infection decreases the transmission of certain viruses like dengue. So they don't really understand why. People are studying this now, but if a mosquito is infected with certain strains of Wolbachia, putting aside the conditional male sterility, The presence of the Wolbachia somehow inhibits the transmission. It might be through the replication of the virus, but it eliminates the transmission of the virus. So even if you were bitten by a mosquito carrying dengue, you'll be less likely to contract the disease or virus. So the idea here is that, let's just say you have an uninfected mosquito population, or it's infected with some strain that doesn't really do too much. It might be a naive strain. But let's just say for now, it's easiest to imagine it's not got Wolbachia in it. And then just imagine that you take and you release some really healthy mosquitoes that are carrying Wolbachia into that population. And let's say that that strain of Wolbachia induces this conditional male sterility that causes the uninfected females to not do so well. So their numbers go down. And because there's a selective advantage to the ones carrying this Wolbachia strain, they sweep through the population. By the end, the goal is that you have completely swept through Wolbachia. Your whole mosquito population has Wolbachia, and then they're less likely to transmit the virus. There's another goal, another thing that could be achieved, which is using Wolbachia to sterilize the mosquitoes. So let's just say you have a mosquito population with some Wolbachia strain. I'll just call it A. And so then you release males, only male mosquitoes that are infected with some other strain of Wolbachia. Now, one thing I didn't say before is this conditional male sterility. I said that the Wolbachia in the egg can, as the anecdote, it can fix the problem in the males so that there is a rescuing effect, but they have to be the same strain. If they're a different strain, that other strain cannot reverse the effects. It can't undo the sterility effect, and there's death. So if you release a bunch of males into a mosquito population and it has a different Wolbachia strain, that male sterility is going to kick in because all those males are just going to swamp all the native males, and they're going to mate with the females, and all their babies are going to die. And eventually you're going to have more and more of those released. So there's a plan for successive release of these males. And over time, you're basically going to cause the death of these mosquitoes. This whole population is just going to crash. Okay. So before, this strategy is not really getting rid of the mosquitoes. It's just eliminating their transmission of this virus. You're just infecting them with Wolbachia. Here, you're really trying to you're just trying to eradicate the mosquitoes with Wolbachia. Believe it or not, there's some controversy here because there are, not necessarily with some of these pest strains of mosquitoes, but there's a fear that there could be some indirect cost to other species of Wolbachia that don't carry human diseases. And mosquitoes. And some mosquitoes actually don't suck blood. They suck nectar from flowers, and they're pollinators. And so, the question is it really okay for us to be eradicating whole species of, number one, whole species of mosquitoes, and number two, there's a risk to things jumping from one strain to another or another species and causing harm. And I just put this slide in here because I have a colleague who's at UC San Diego, and he works in it's a relatively new mosquito unit in the genetics department. And he is working with the French Polynesian government to take an island. This is not his island, but he has been designated an island in French Polynesia to release mosquitoes for these mosquito control pilot experiments. Other locations, this is not just the only place it's being done. It's being done in Singapore, Thailand, Mexico, and Australia. There's been a lot of Bill and Melinda Gates funding that's gone into spearheading these projects. Okay, I'm almost done, but the last thing I'll just say is recently, I'd say in the past year or so, year and a half, it has been discovered that there are two genes that cause cytoplasmic incompatibility that Wolbachia uses, that they express to cause that conditional male sterility. And, the names are not really that interesting. They're called CIF-A and CIF-B. So here they are. And the idea is that the Wolbachia in the male, let's just say this is an infected male, The Wolbachia expresses these two genes, and these two genes actually will poison the sperm's hereditary material. So that's why I've highlighted it red. So the sperm that's toxified by these two genes will fertilize an egg from a female that does not have Wolbachia, and that causes the destruction of that half of the genome, and that embryo dies. However, if, and they know this because they've done experiments expressing these genes with transgenics, so no Wolbachia, they just do all of this work with just the genes. If you express the CIF-A gene alone in the egg during development, you get fertilization, and that CIF-A gene acts as an antidote to undo the damage that's been done to the hereditary material of the sperm, and that embryo lives. So that's kind of where the field is right now. Is there are a lot of groups in different places that are really interested in understanding how these bacteria, whatever they're doing, whether it's parthenogenesis or feminization or conditional male sterility, what are the genes that these bacteria are using to cause these effects? Because those could be tools for manipulating these insects. And I'll just end this is my last slide, but just to say, there's a prediction, and it's not just a prediction now, it's actually groups are ramping up to do this, is the bacterial genes that cause these reproductive manipulations are going to be used to actually manipulate a lot, not just mosquitoes, but a lot of other pests. And that really bypasses the need for bacteria. You don't have to work with the bacteria anymore. You can just express these genes through some trickery in these insects and release those insects and get the same thing done. So with insects like the tsetse fly, the sand fly, which causes leishmaniasis, this is an insect that causes Chagas disease. And then all of these on the bottom are pests of crops and whatnot. There are a lot of targets, and this is kind of I think the wave of the future in terms of pest control. And that's it.